work great. They just give you different outputs. Here you're not making nulls, you're turning transcription down. Here you make true nulls, but you also have heterogeneity in protein expression across the population. And Bruce's lab showed that this system in general, as in cancer models, works quite robustly to control transcription of a variety of types of endogenous genes. So just qPCR data. But overall it was a nice proof of principle set of experiments showing that we could turn off transcription in iPS cells in an inducible way. And so that you can imagine this is especially important for studying differentiation, where you might want to grow your iPS cells up, infect in a guide, induce some differentiation protocol, for example, that guides you down a neuronal lineage or a cardiac lineage, and then at a specific time point during that differentiation process, turn off a gene and ask how is that gene required for neural differentiation or cardiac differentiation. And so that's all possible with either of these systems, really, in most ways, but definitely we're able to control transcription in iPS cells. One of the things we noticed really early on that I like to reemphasize in this workshop is that in many settings in human cells, expression of an sgRNA is the limiting factor for a lot of types of experiments, whether it's imaging or, in many cases, editing, if you're not using RNPs, or for controlling transcription. So one of the ways we saw this was that we were knocking down these GFP reporters, and we saw that the level of knockdown, so this is expression of a GFP reporter, was to some degree correlated with indirect measurements of guide expression. So the more sgRNA you express, the more you repress this GFP reporter. And so that was a problem for us early on, because we had an eye towards doing functional genomics where we perturb expression of all genes encoded in the genome. But to do that, you need to build a platform that relies on guide expression from a lentivirus that's integrated at single copy into the genome. And so if we had a dose-limiting expression of our sgRNA, that wasn't a very satisfying position to start from for building a functional genomics platform. And so together with Bo Huang's lab and Stanley Chi, we tested a series of changes to the original sgRNA design. So this is the original published design from Jennifer that was published early on. And so actually a modified form of the natural Pyogenes hairpin that's a bit truncated. So it's truncated by 5 bases. So we tested a number of things, but we tested just what happens if we just extend the hairpin to the wild type length, and that seemed to be more active, as these were the active changes that we made. And then the other thing we noticed is that there's a Pol3 terminator in the stem of the Pyogenes tracker RNA. So if we remove that stretch of 4 T's and just break it up with an A, you increase expression of the guide RNA by about 5-fold, which is beneficial in many cases. And then if we combine both this change that stabilizes the sgRNA structure along with the change that increases transcription, what we saw was that we got a lot better activity when we were trying to control transcription with single copy lentiviral integrations of the guide. So if this isn't always affected, if you're doing transient transfection experiments, often expression off of plasmid is really robust, and your experiments won't be limited by either the expression of the Cas9 or Dcas9 proteins or the guide. They're just more limited by other factors. But we and others have seen that this guide design in our hands works a lot better. And just so you don't think I'm only talking about our work, this is one example of another lab from Texas, I think, that showed the same thing. And at this point, there are 4 or 5 other papers that have shown that this modified guide design is generally better than what most people are using. So it's a bit, I'll get off my soapbox, but basically it's a bit surprising that people haven't switched toward this, because there's no consequence, as far as we can tell, to having, well, unless you want an inducible system, system like Dana's using. But in many cases, you want to maximize your guide expression, as these changes help with that. So there are a variety of tools for silencing transcription or doing loss of function experiments, including RNAi and CRISPR, now CRISPR-I in human cells. So we were excited about additional functions that we could specify using dCAS9. So we tested whether we could activate transcription or as well. You can imagine this would have a variety of applications for gain-of-function experiments that could be useful in studying cancer biology or development. And so, again, we took a page from the artificial transcription factor field and used the sort of who's who of domains to try to activate transcription of reporters. We fused this VP16 domain in 4 copies to dCAS9, and then targeted a GFP reporter and asked then this reporter importantly has 3 binding sites for the sgRNA we're expressing, and asked whether we could turn GFP expression on. So if you look a couple days later by flow cytometry, we can see that specifically when we co-express the dCAS9 activator protein together with a guide targeting this promoter, we turn on GFP expression. So this is proof at least that we can fuse these domains to dCAS9 and use them to target the genome and activate transcription of reporters. Disappointingly, when we started to try to test our ability to activate transcription of endogenous genes, we saw very quickly that this scheme doesn't really work very robustly to activate endogenous gene expression. This is not entirely surprising. Yesterday I showed you a slide from Keith Jung's lab where they were using tail VP64 fusions, and I didn't really walk you through the slide, but essentially what the point of the paper was that one tail VP64 fusion is usually not enough to activate transcription of an endogenous gene. And they saw that with tails you had to have 4 adjacent or 3 or 4 adjacent binding events to get activation of most endogenous genes. So that's essentially the exact same thing we're seeing here, where if you have 3 adjacent binding events, you get nice activation of a gene, where if you have a single binding site, you're only recruiting 1 copy of this VP64 domain, you don't get much activation. And so at that point, I think the field in general started to engage with this idea that a simple artificial transcription factor that activates transcription with a single binding event was not going to be enough, or at least not enough with a single activation domain. And so we and others started to search for additional ways to recruit more activation domains with the expression of a single sgRNA. The way we solved this problem was we fused a highly optimized epitope tag to dCAS9. So this is derived from a yeast GCN4 protein. It's a fairly linear epitope that can be connected by a short flexible linker. So we fused 10 copies of this epitope to dCAS9. And so now in theory, with expression of 1 sgRNA, if we co-express a single chain variable fragment fused to this same activation domain, we can recruit up to 10 copies of the activation domain, sort of using a single sgRNA with a version 2 system to amplify our signal. And we showed in human cells for a variety of endogenous genes that this really worked much better than the version 1 system. So here we're looking at just a test reporter gene called CXCR4. The gene's not really important, although for the aficionados this is a HIV co-receptor. And you see with our version 1 system, when we target CXCR4, we really don't activate transcription at all. If you use our version 2 system, where we're now recruiting 10 copies of this activation domain, we see we turn on gene expression by about 40-fold. This gene is pretty much off in these cell types, so it's difficult to normalize a fold change. If you subtract background fluorescence, you can get artificially large fold changes, and so the field struggled a little bit with how to display these kinds of data. What I like a lot better is just to display the primary flow cytometry data. So these are K562 cells that are unstained, so this is autofluorescence. If you stain them for CXCR4 expression and in cells that express a negative control guide RNA, you see they look the same as the unstained cells, indicating there's really almost no CXCR4 expressed on this cell type. If you then activate CXCR4 expression by expressing a guide in the presence of this version 2 CRISPR-A system, we see that most of the cells in the population activate expression, and that it's fairly homogeneous across the population. And so our ability to, there's a little bit of a subtle point, but sorry. answer the second question first. No, we don't. For the most part, we don't see positional effects where you activate neighbor genes. If you have two anti-parallel promoters that are very close together, you'll activate both of them. But the window in which we activate transcription is quite narrow. It's only about 1 to 2 kb. So the transcription start sites for endogenous genes are generally more spaced out. There are caveats to that answer. There are a lot of described non-coding RNAs that are anti-parallel to coding genes and are very, very close with the with their divergent transcription start sites. And so we can't really decouple the function of an adjacent non-coding RNA, although that's a bit of a tricky thing to say, because often the expression of those anti-parallel non-coding RNAs is intimately also involved in regulation of the coding gene. So you might need additional methods to dissect those. For the first question, that's sort of hard to measure. The affinity between the epitope that we're using and the scFv is extraordinarily high. I would be surprised if you didn't have the scFv bound to dCas9 constitutively, and that as we haven't measured it. If you do, like we've done some FRAP experiments where we're just imaging telomeres using this same setup, and I don't know, that doesn't really tell you the answer to your question. I don't know. So like I said, the field was searching for a series of second-generation CRISPR-A constructs. There have been quite a few published at this point from us and Ron Vale, from George Church's lab, from Feng Zhang's lab, and Janish's lab, and who else is on here? Charles Gershbach, maybe Stanley. And anyways, there are at least 8 now published. And George Church did the field a service by comparing them all in a Nature Methods paper described here last year. And I have a few issues with the way he did this, but we'll just take him at his word. And he says that ours, this Viper system, and the SAM system from Feng are the best. All 3 sort of convergently arrived at the same idea that you need to use multiple activation domains to robustly control transcription of, or robustly activate transcription of endogenous genes. And so you can read about these, they're all published, but they all converge on the same idea, essentially. So I want to introduce an idea that's been really, really useful over the years in synthetic biology. So far today I've talked predominantly about controlling transcription or doing a variety of things by fusing proteins together artificially. Another way that you can control protein localization is to use RNA aptamers that have high affinity for proteins. So a lot of these are depicted here are a bunch of RNA stem loops. Most of these are derived from phage viruses, and these have been used or have been characterized since the 70s or 80s as having very, very high affinity for specific phage proteins. So they're involved in phage genome and viral assembly, and so these are often coat proteins that coat the viral phage genome and are bound very tightly. And so there's quite a number of them actually. There are 6 listed here. This top one, MS2, is perhaps the most famous one, but we can see is that this MS2 coat protein derived from a phage binds the MS2 stem loop with a very, very high affinity. So this is the KD listed here. And so this has been used in the past in synthetic biology to control recruitment of proteins using an RNA rather than using a protein. So we're talking about RNA-protein interactions, not protein-protein interactions. And so one of the ways in which a second-generation CRISPR-A system for activating transcription was derived was to take advantage of the fact that we can append these MS2 stem loops onto an sgRNA. So now, rather than dCas9 recruiting activator domains, the RNA is recruiting the activator domain by interacting with a second protein that's expressed. And so Feng appended in his CRISPR-A system VP64 domain directly to dCas9, and then he also appended 2 MS2 stem loops internally on the sgRNA in a manner that was informed by the crystal structure. So this was like really nice piece of work. They waited until they had the crystal structure and then didn't just blindly stick these MS2 stems onto the sgRNA sort of a unguided fashion like we did, but instead said we're going to use the structure to figure out where we can append these and not impact the function of the sgRNA. And stuck to there, then showed that this doesn't compromise the activity of sgRNA loading into Cas9 or dCas9. If you co-express an MCP coat protein that binds to these MS2 stem loops, fused to 2 activation domains called P65 domain, this is derived from the endogenous NF-kappa B pathway in human cells, along with the HSF1 activation domain, which is also an endogenous activation domain. You're now with a single sgRNA recruiting 3 different activation domains. So this one's recruited with the protein, and these 2 are recruited by the RNA. And he showed in this paper that this works quite nicely to activate transcription of endogenous genes, and it is another way to multiplex your ability to deliver different domains using a dCas9 and a single sgRNA. We had a similar idea at UCSF. We didn't have the crystal structure at the time, so we just appended these stem loops to the C-terminus of an sgRNA. This is work from Jesse Zalatan, who has his own lab at University of Washington now, but the work was done in Wendell Lim's lab at UCSF. And what he was able to show is that, as I've shown you previously, we can use dCas9 to control transcription, either just by using dCas9 alone or by appending protein domains to dCas9. And in this setting, the guide RNA that you express only encodes the locus that you're targeting. It doesn't do anything functionally. In contrast, if you start to append these RNA domains to an sgRNA, now the guide RNA encodes both the locus that you want to target and the action that you want to perform. So you can append different hairpin structures to an sgRNA that are targeted to different genomic locations, and with one dCas9 will activate and repress transcription at different loci. So that's shown here, where you have this purple stem loop that recruits this purple protein fused to this green effector domain, and that's controlled by this great dCas9, so this can all be targeted to gene X. And then in the same cell with the same dCas9, you don't need an orthogonal dCas9, you can append a different sgRNA with a different stem loop that recruits a different protein that's fused to a different effector domain to do something different at a different locus. So it just increases your ability to use one CRISPR system to control the expression of multiple genes in different ways, in a way that you can imagine could be really useful for a lot of applications. And so the question was, does this work? Jesse fused 3 different stem loops to the sgRNA and showed that each of these, when you co-express protein that recognizes a stem loop fused to an effector domain, can activate transcription of a reporter gene in yeast. So they're quantitatively a bit distinct, but you can see that if you co-express all these components together, each of these will activate expression of this Venus reporter. This can be used to, he showed that there was no crosstalk between binding pairs, so you can co-express these in the same cell, and they don't encode the wrong functions at the wrong loci, so another way of saying that is they're completely orthogonal. You can use this same sgRNA scaffold general structure to recruit, to amplify your signal. So you can append multiple stem loops to one sgRNA to either recruit 1, 2, or 3 of these effector proteins. And that can have an additive effect on transcription. And then you can also append 2 different RNA stem loops to the same sgRNA. So here we're appending an MS2 stem loop and a PP7 to the same sgRNA, and in this manner you can recruit either 2 of the same domain using an RNA, or 2 different domains using one RNA to control transcription. This also works in human cells, so we used the MS2 system, or this COM-CRAB system, to show that in human cells with one dCas9, we can control expression of 2 genes and turn one off and turn the other on. And so That's shown here, where here we're activating expression of CXCR4, or we're turning off expression of B4-GALA-T1, or we can turn on one gene and turn off the other gene at the same time using one dCas9 scheme. So it just increases your flexibility for controlling transcription across the genome. This works for metabolic control, which you could imagine, like the ability to control multiple genes dynamically could be really useful for metabolic imaging. And so in yeast, Jesse was able to show that he could tune the output of this violacin pathway. This is a fairly complex pathway that has, it's a 5-gene pathway, so violacin A through D, and depending on which gene is activated, you get production of these different metabolites that have different colors. So this one's pinkish, that one's purple, yellowish, and sort of a cyan. And so what Jesse was interested in is whether you could use a single dCas9 to turn off and on different steps in the pathway and tune bioproduction of these different metabolites. So this works, and he showed that in the absence of any sgRNAs, you get this, the pathway is off, more or less. If you start to activate the pathway, you produce this PV intermediate. If you activate the first 2 steps, you produce V. If you activate this first step and turn off D, you now produce PDV, which is shown on these HPLC curves as different peaks for these metabolic intermediates. And then if you activate the first 2 steps and turn off the last step, rather than producing this, you produce that. And so sort of a toy experiment, but nice ability to show that we can control expression of more than one gene to specify the output of a pathway in a organism that is useful. So I think some of the questions remain at this point, and some of these will be answered by Mike, are can we manipulate pathways in the same way that I just showed you for yeast and human cells? Can we use CRISPR-RNA to control expression of all the genes encoded in the genome and that way build a functional genomics platform? I sort of already showed you this, which is can we use CRISPR-RNA to control the expression of non-coding RNAs and try to figure out the expression of many non-coding RNAs that are annotated in mammalian genomes? It's hard to study a non-coding RNA with Cas9 because non-coding RNAs don't have an open reading frame, so there's nothing really to disrupt in the same way that most people are doing genome editing. If you want to disrupt a non-coding RNA with Cas9, you have to delete a large region of the genome. That's less efficient and also can have unintended consequences if you delete additional regulatory regions. So we think that CRISPR is like a very surgical way to just turn off transcription without disrupting large chunks of the genome, and it's useful for studying non-coding RNAs. And then we'd like to start to move towards higher perturbations. the chromatin marks at that locus dictate transcription. We've used, sort of retrospective techniques like ChIP-seq, where you mix a cell, you pull things out, and you have to correlate the presence of different marks with different transcription statuses in human cells. What we'd really like to be able to do is take a living human cell, edit a mark at a specific site, and ask how that turns transcription on dynamically over time, like through differentiation or through some drug treatment. And so labs have started to do this. So building off of sort of our initial work in yeast and human cells, people have used dCas9 and appended on additional domains. And so some of this work is really all from 2016, where people a couple different groups have fused on DNA demethylases to remove methyl marks off of DNA. So I talked entirely about histone marks, but DNA also has epigenetic changes. Just methylation, sorry, not changes, that can be dynamically deposited or removed to control transcription. And so to remove or deposit DNA, people have created dCas9 demethylase fusions and dCas9 methyltransferase fusions. People have also fused on histone acetyltransferases and histone demethylases to edit the marks on histones. So I'll just talk you through some of this very briefly. One of the first papers was from Renee Mayer's lab at UMass, published in 2015, where they fused, actually, this is Charles Gershbach's lab, not Renee Mayer's lab, sorry, backwards. They fused the P300 histone acetyltransferase domain to dCas9 and asked whether they could deposit acetylation marks on an endogenous gene using by targeting dCas9 fused to P300 domain to that locus. And so they showed this, that they could deposit acetylation marks by ChIP-seq, but also just asked whether deposition of increased acetylation would activate transcription. So that's shown here, and you can see that they can quite robustly use these dCas9 P300 domains to deposit acetylation at these three endogenous genes, and that turns on gene expression. So rather than, like we had shown previously, use scaffolds, now they're directly editing the epigenome to activate transcription. One of the coolest things I think about moving beyond scaffolds towards actual epigenetic enzymes is that we can control the chromatin or DNA epigenetic marks at regions very far from genes, so distal enhancers, and this has dramatic effects on gene expression. So this is really the first example of how this might be done using CRISPR. So here we have a really classic locus in the human genome. This is the hemoglobin locus, so it encodes the different hemoglobin genes that are expressed at different times in your life. So we have fetal hemoglobin and adult hemoglobin and whatnot. They're all encoded at the same locus, so that's shown in these white boxes. There's this classic hemoglobin enhancer that's upstream of these genes, and what the Gershbach lab was able to show is that if you target this P300 domain to the enhancer, you get activation of gene expression very distally. So we're depositing acetylation marks at an enhancer, which activates transcription of genes that are up to, however many kb that is away, almost 100 kb away. So this is thought to be mostly interacted by the 3D structure of the enhancer interacting with these genes, but was proof of principle that you could use these tools both to activate transcription and putatively to annotate functional enhancers across the genome by depositing or removing specific chromatin marks. And they did all of the right controls. If you're interested in this, this is published, but basically they showed if you look at these enhancers or the proximal promoters for these different genes, with the dCas9 BP64 domain, you deposit H3K27 acetylation where you target the dCas9 fusion, but not at distal sites. So that's shown by these ChIP experiments here. But in contrast, if you target this P300 domain to the enhancer, you see acetylation marks that are deposited very distantly from the genomic location in 2D that you're targeting the dCas9 P300.